It's 10:15, so I think we're going to go ahead and start the session. Welcome to Symposium 32, Gene Delivery, Targeted Nonviral Delivery to Airway Epithelial Cells. My name is Piotrowski-Daspit. I'm an assistant professor of biomedical engineering and internal medicine at the University of Michigan. And just a couple quick housekeeping things. So on the app, you can submit questions online, but we'll also, of course, take questions live here as well. And then I'll turn it over to my co-chair. Introduce
Thanks, Alex. My name's Steve Hart. I'm from University College London, and it's great to be here. Hope you've all been having a great conference. So we're going to have four speakers in this session, and there will be questions after each talk. But after the fourth, there's going to be a short panel session, about 15 minutes. So please store up your questions and comments for discussion then. So without any further ado, I'd like to invite the first speaker, Dan Segut, to the podium. Thanks.
That's good. It'll keep me honest. Yeah. Thanks. Wonderful to be here, and good morning to everybody. Today I'm going to talk about targeting multiple tissues with non-viral lipid-based nanoparticles as a way to introduce the possibilities of correcting CFTR in places beyond the lungs someday in the future. These are my conflict of interest disclosures. Where I want to begin is in the vast promise of genome correction strategies. Right now, a company, Intellia Therapeutics, is finishing a phase 3 clinical trial wherein they're administering a single injection of a lipid nanoparticle that co-delivers Cas9, messenger RNA, and a single guide RNA designed to knock out this protein transthyretin that causes TTR-mediated amyloidosis in some individuals. This is a case whereby elimination of the protein is therapeutically beneficial. As you can see here in humans, this single administration eliminates this protein to about 10% of the original level, and it's durable and persistent. Now, in cystic fibrosis, there are additional challenges. We would need to correct the mutation, not eliminate it. We would ideally do this in multiple tissues, starting with the lungs. We have found in this last couple of years that editing in the lungs is likely durable as well, which I think is great news for, again, the future 5 years, 10 years, therapeutic strategies by which one may be able to correct CFTR in human lungs and see durable, long-lasting physiological function recovery. The way that this can be achieved is by targeted nanoparticle development. This could be functionalizing with antibodies, could be physical chemistry. In the case of what I'll talk about today, these nanoparticles utilize a mechanism of action called endogenous targeting, whereby they absorb plasma proteins from the blood to redirect to different tissues and utilize cell surface receptors in hepatic and extrahepatic cells. So we discovered that when taking a traditional 4-component lipid nanoparticle, if these LNPs could be augmented with a supplemental 5th molecule, and if this molecule contains a quaternary ammonium head group and is introduced into the nanoparticle at 40 to 60 mol%, these LNPs will avoid the liver and accumulate in the lungs, where they transfect lungs from the endothelium to the epithelium. Conversely, if one incorporates an anionic lipid with a carboxylic acid or a phosphate head group, these nanoparticles will now internalize into cells in the spleen, including T cells and B cells, and this should be incorporated at 3 to 30 mol%. And the key molecules for binding apolipoprotein E for introduction to hepatocytes by the LDL receptor, are these ionizable aminolipids, and this is the path forward to enrich delivery across the liver. So what I thought I would do today is introduce this additional genetic disease, alpha-1 antitrypsin disease, as a case study to imagine how this kind of technology or strategy across viral and non-viral nanoparticles could be relevant in cystic fibrosis. This disease is one that affects both the liver and the lungs, so we contemplated whether it would be possible to inject mixtures of nanoparticles, one that goes to the lungs, one that goes to the liver, and achieve targeted correction in multiple tissues. This disease is caused by mutations in the SERPINA1 gene. These mutations lead to multiple issues. So first, the mutant alpha-1 antitrypsin can aggregate in the liver. This aggregation causes liver damage. Second, the secreted alpha-1 antitrypsin no longer has the neutrophil elastase function in the lungs. So you have this dual gain of function, loss of function mechanism happening, whereby there's an aggregation-induced problem in the liver, as well as a loss of enzyme activity in the lungs from the secreted misfolded protein. What's attracted us to this potential problem is that about 70% of all AAT from the literature is made in the liver, meaning that 30% is likely made somewhere else in the body. And in addition, only 5 to 10% of reported AAT can diffuse into the lungs, meaning that even that massive amount, 70% that's made in the liver, not all of it reaches the lungs. So this set up a challenge, which is, and a question, a fundamental biology question. Could lung cells contribute to a local increase in alpha-1 antitrypsin levels in the lungs? I've been really struck at this particular conference, particularly in the poster sessions, concerning the involvement of lots of different cell types for cystic fibrosis. Of course, we're always focused on the lungs, but I learned about a variety of different cell types in the lungs that contribute to this disease. And I think this is the kind of path forward that we may be able to employ for CF correction. So first, the postdoc who worked on this project, Minjung Kim, we aim to optimize the liver targeting sort LNPs to further improve their ability to deliver long base editor correction RNAs. So we did this by selecting a series of supplemental ionizable aminolipids with the chemical structures shown here. We systematically evaluated incorporation of these lipids with the goal to increase liver transfection to improve the gene correction potential in the liver. And we identified this lipid shown here, 4A3-CYT, which has a citronellyle motif that improves endosomal escape. So those saturated bonds and the motifs here with branching at the chain ends of the hydrophobic domain increase the endosomal escape, which leads to increased transfection of liver hepatocytes. This fivefold increase in transfection of the liver could then be utilized in our therapeutic models in the PiZ mice. We evaluated which cell types internalized and functionally delivered the mRNA through utilization of the AI14 mouse. This mouse contains a lock-stop-locks TD tomato cassette in all tissues. Successful delivery of Cre recombinase DNA leads to DNA recombination in those cells and turns on red fluorescence in any transfected cell population. This is a light switch, if you will, and allows us to determine and quantify which cell types are transfected. So interestingly, we found that the liver sort LNPs transfected a variety of cells in the liver, including Kupffer cells, endothelial cells, and hepatocytes. And the addition of this 4A3-CYT to make the 5-component LNP increased transfection of hepatocytes, which was the targeted goal of this development. Next, we focused on lung sort, again with the vision to combine lung sort and liver sort in a dual sort strategy. And in this way, we've started with the canonical lipid that's been used for decades, DOTAP. We've recently synthesized over 450 analogs, as well as evaluated the 12 known similar analogs that are commercially available to identify molecules that are more potent and less toxic than DOTAP. For the purposes of this presentation, we screened a series of TAP and EPC-related lipids and identified this molecule abbreviated DORI, whose chemical structure is shown here. Incorporation of DORI as a substitute for DOTAP increased the efficacy in the lungs with reduced toxicity, potentially because that quaternary nitrogen is no longer at the chain end of the molecule, but is buried a little bit in the center of the chemical structure. So here we selected the ionizable lipid 4A3-SC7 mixed with DOPE, cholesterol, PEG-DMG, and supplemented it with 40 mol% of DORI to promote lung delivery. So with this particular formulation and back in the AI14 mice, we observed a high transfection of endothelial cells and interestingly about 20 to 25% transfection of AT2 cells in the lungs, which was the target cell for this particular disease manifestation. In addition to lipid nanoparticle development, one can optimize the genome editors themselves. In this particular example, we are using adenine base editors to target the mutation that's shown in the first slide for SERPINA1 to correct it back to the wild type sequence. With a base editing strategy, there are multiple angles to optimize the particular constructs. You could optimize the deaminase, the Cas enzyme, the PAM site, the guide RNA, and the chemical modifications. So Minjung first examined a set of different adenine base editors, and in this case we utilized human liver fibroblast cells that contain the human mutation. So it's kind of the corollary to the human bronchial epithelial cells in CF research. The mutations are shown here, and we examined 3 different adenine base editors, the ABE8E NGC, NCAC ABE variant 9, and ABE8E NG that has a shorter PAM. As shown in the graph on the right, Minjung found that the ABE8E NGC adenine base editor led to the highest conversion of AT to CG, which was able to make the correct wild-type sequence at about 17%. Next, we varied the length of the single guide RNA and explored different photospacer lengths. We compared 18, 19, and 20 nucleotide single guide RNAs and determined that the 19 nucleotide single guide RNA led to the highest level of correction in these human fibroblast cells. With that in hand, we aim to further optimize delivery of the long adenine base editor mRNA. These mRNAs are 3 to 4 times longer than conventional reporter RNAs like luciferase, and we and others have found that it's necessary to optimize the lipid chemistry and the ratio of lipids to incorporate these significantly longer RNAs into the nanoparticle. The nanoparticles are prepared by mixing all lipids in an ethanol phase and rapidly mixing with an acidic solution of the cargos in nucleic acid format. We compared delivery of all-in-one LNPs that would contain both ABE mRNA and single guide RNA, and LNPs made that have only the mRNA and only the guide RNA to determine whether it would be possible to formulate all together or be necessary to deliver 2 different LNPs that contain different payloads. Interestingly, when we examined the encapsulation efficiency, we found that both the all-in-one and the mRNA-only LNP had a significant loss in encapsulation efficiency down to 50%. Conversely, the guide RNA was able to readily load into the LNP, likely due to its significantly smaller size of only about 100 nucleotides. So this meant that the long mRNA was really the problem, and it set out an engineering optimization opportunity to improve this co-loading strategy. So Minjung took our initial formulation and then adjusted the molar ratio of these lipids. For example, we increased or decreased cationic lipid or phospholipid or pegylated lipid in a design of experiment workflow. These kind of studies are difficult to explain with presentations, so I refer you to the slide, to the paper that we recently published. At the end of the day, through comparing in vitro and in vivo all of these different combinations, Minjung found that a subtle change to the ionizable lipid and the phospholipid allowed for a dramatic increase in the assembly of the long ABE mRNA into the nanoparticle, whereby now 87% of total nucleic acid could be encapsulated with biophysical properties appropriate for delivery to the liver or the lungs, about 75 nanometers with low polydispersity index. So this then allowed us to examine both the optimized lung-targeting LNP and the optimized liver-targeted LNP in the context of a therapeutically relevant mouse model of alpha-1 antitrypsin disease. This PIZ model, we thought, could allow us to compare and contrast potential benefit against the toxic gain-of-function mutation that occurs in the liver versus the loss-of-function mutation effect that occurs in the lungs. So first, examining effects in the liver, Minjung intravenously administered the top-performing LNPs from the optimization campaigns, and we compared side-by-side liver sort LNPs, dual sort LNPs, whereby the animals received both nanoparticles, or lung sort LNPs, whereby they received only the lung-targeting nanoparticle. The injection campaign is shown in the top right. We then isolated DNA from these tissues and performed next-generation sequencing to quantify the levels of correction in the tissues. As shown here, in the liver, both the liver sort and dual sort LNPs led to 30 to 40% correction of SERPINA1, and lung sort LNP, as expected, led to little to no correction in the liver, although there was correction in the lungs at about 10% of both dual sort and lung sort LNP. We further examined the durability of these correction events in this mouse model of alpha-1 antitrypsin disease. The colors here are designating the different sort LNP administrations. Liver sort's blue, dual sort's yellow, and lung sort is pink. The green bars are showing the correction editing, which is the sum of 7G and 5G plus 7G mutation corrections, both of which are therapeutically beneficial, whereby we see durability in the liver for both liver sort and dual sort LNPs out to 32 weeks after the injections. And the problem for the alpha-1 antitrypsin mutation is this aggregation effect that occurs in the liver. So using staining, PAS-D staining, to determine and quantify the globules that aggregate in the liver, we're able to quantify a significant reduction of about 40% for both liver sort and dual sort treated animals. By quantifying the PAS-D positive area staining, we see that there's no change when correcting only the lungs in the liver phenotype, but there's a significant change and improvement in the function for both liver sort and dual sort treated animals. This protein, the alpha-1 antitrypsin protein, is secreted. So we next wanted to examine whether correction in the liver or lungs could promote secretion and circulation of the functional enzyme in the blood. To do that, we drew blood from IV-administered animals in these different groups, liver sort, dual sort, and lung sort treated animals. Using LC-MS, we quantified the correct isoform of the alpha-1 antitrypsin enzyme in the blood at different time points, 4 weeks, 12 weeks, and 32 weeks. As shown here, the corrected functional alpha-1 antitrypsin was now quantifiable in the blood for all of the liver sort and dual sort treated animals. Although we did not see a significant improvement in the secreted functional alpha-1 antitrypsin for the lung sort treated animals alone, suggesting that this protein may be localized and contained in the lung environment. Then finally, we addressed the toxic loss of function mutation in the lungs, and this is where things get a little interesting and hopefully have some relevance to cystic fibrosis. So here we compared dual sort LNP injection, whereby we would have these animals administered with liver sort or dual sort LNP to determine if there could be benefit from correction of the SERPINA1 mutation in the lungs. We administered these animals and euthanized them 6 days after IV injection, and then used next-generation sequencing to quantify the corrected SERPINA1 in the lungs and the liver. So in the liver, we found that both liver sort treated animals and dual sort treated animals both had 15 to 20% correction of SERPINA1 in the livers. But interestingly, only the dual sort LNP had correction in the lungs, and that level was about 10%. So now we could isolate the bronchial lavage fluid from these treated animals to ask whether there could be local benefit from correction of SERPINA1 in the lungs. And as shown here, when we measured the protein levels in the BALF, we saw a significant increase from 10 to 17 micrograms per milliliter of the corrected enzyme in the lungs, indicating that indeed the lungs are producing and secreting this protein, and the treatment of the lungs can generate secretion and production in the lung environment better than treating the liver alone. And on the right-hand side, we did a functional study whereby we measured the relative elastase inhibition to measure the function of this enzyme, and found that indeed correction in the lungs with dual sort LNP improved the elastase inhibition significantly from 70 to 90%, again suggesting that there are cells in the lungs, primarily AT2 cells, that produce and secrete this protein, and correction in the lungs has added therapeutic benefit. So I feel the broader impact could be in cystic fibrosis as we contemplate correction of this protein not only in the lungs, but in the liver, in the intestines, in the pancreas, whereby there are local phenotypes that could be alleviated and symptoms of the disease that could be alleviated by this long-term goal of developing nanoparticle AAV stem cells and other regenerative therapies that may address the manifestations of CF disease in multiple tissues. And interestingly, in this study, we did find that correction at the local level in the lungs, even though the liver is thought to be the primary generator and secretor of this enzyme, does have a significant enhanced beneficial effect in the lungs. So with that, I'll finish and acknowledge again Min-Jung Kim, who's now a professor in South Korea, and other investigators here and collaborators who helped with this particular study, and be delighted to take questions.
Amazing, thank you. All right, so please feel free to come up to the microphones, but I can start us off. So I have 2 questions. I'll start with one. So in your nanoparticle optimization, when you're going from sort of the all-in-one to sort of multiple cargoes, it seemed like size was really important, right? So in your all-in-one formulation, it was, almost double the size, right? So it's 130 nanometers compared to 75 nanometers in your lead formulation. So what's really driving that change in size? Is it, the lead formulation has components that are just better able to condense everything, or what are your thinking?
Yeah, LNPs were initially developed for short interfering RNAs, siRNAs, and those are 18 to 22 base pairs. So it's possible for hundreds of siRNAs to fit inside of one lipid nanoparticle. However, mRNAs, from reporter contexts, say 2,000 nucleotides, to these base editors, which are 5, 6, 7,000 nucleotides, it's been shown that only between 2 and 8 mRNA molecules can fit inside of each messenger RNA. So there are elements of forming those water pockets and convincing the long mRNAs to assemble into the LNP and trying to get as many copies as possible into each lipid nanoparticle composition. And this is done through the lipid optimization campaign. I think it matters too from a dosing perspective, because we always dose drugs from a mass benefit. So if you compare just the luciferase to these base editors, there's about 3 times more mRNA molecules for a luciferase at the same mass of drug that's administered into the animals. So you're kind of diminishing the returns, and things just get harder and harder as you go up in the size of these cargoes. Yeah.
Okay, great talk. Thank you very much. So I have 3 questions. The first one is regarding all salt LNPs. How is the stability of salt LNPs, and have you studied the in vivo immunogenicity post-IV infusion in the animal models?
Yeah, the sort LNP contain additional lipids. So the first case, the lung sort I showed as a cationic lipid, these have very high stability because they maintain the electrostatic interaction with negative RNA. The spleen sort LNPs have lower stability because they incorporate negative lipids that can repel the negatively charged RNA. So I refer people to the Nature Protocols article that we wrote, where we decreased the amount of the ionizable anionic lipid to 10%, and that's a percentage that still tolerates high loading of mRNA. And then the immunogenicity relates a lot to all of these individual lipids. So it's a very case-by-case study. Jake Brenner runs a lab at University of Pennsylvania who's been systematically comparing the inflammation of different lipids. And the least inflammatory is the one I presented, this 4A3SC8 or 4A3SC7. So it's a real problem, and I think we have to treat it just in drug development terms, individual molecule by individual molecule.
Okay, so in the case of long salt LNP after IV infusion, have you compared your cytokine profiles to the benchmark, such as Intellia's and Analab's?
Yeah, we've not published a lot of that data, but in some of our papers we've examined the effects of delivery of these. So we look at the histology, we look at some of the cytokines and chemokines, and again, it's a very case-by-case study, so hard to answer.
Okay, good. Mm-hmm. So my following 2 questions are regarding the long salt LNP.
Just one, please.
Oh, sorry. Okay. Oh, yeah. Okay, so I'll discuss afterwards. Okay, so the endothelial cells really are the most abundant cells in the lung, right?
Yeah.
So besides flow cytometry, have you examined the cellular localization by co-immunostaining? And also for the clinical perspective, and do you propose to use IV infusion for treatment of CF patients using your salt LNP?
Yeah, I think it's conceivable to use nebulized aerosol delivery or IV delivery, and both strategies have good potential for treatment of CF disease in the lungs, for example. And then in terms of the cell types, we've used immunohistochemistry staining, and recently we've employed single cell sequencing to more accurately and definitively define the cell types, and that'll be coming in a future publication. But we see all of the cells that I've talked about are present in the both mice and rats, is the two species that we've examined so far.
Thank you.
Martin.
Then very nice work. I in particular like sort of the fact that you're looking to get away from DOTAP and then also thinking multi-organ, because obviously you illustrated with AAT, but it's even more relevant for CF, where we have probably more impacted different tissues. What I'm wondering now is, you do this dual sort. Do you see any advantage in like sort of thinking of sequential injections? You do a liver sort first and then a lung sort second. Is there in development or anything like an advantage to inject everything at once versus sequentially?
Yeah, we found that the optimal route is shown here. So we do the liver sort first and then follow it with the lung sort. So we're still learning about the intricacies of that, because the liver will take, almost all the liver cells will take up lipid nanoparticles. So make a long story short, I know we're at time, but it was better to do liver first and lungs second than to do lungs first and liver second. And we're still evaluating why that is. Yeah.
Thank you so much. So our next speaker is Gaurav Sahat.
All right. Let me get started. I have some bad news for you guys. I also have lipid nanoparticles that go to the lung. And as in this conference, I've seen my nanoparticle is better than your nanoparticle. Anyway, so there has been a lot of interest in lipid nanoparticles, rightly so. And these are my conflicts of interest. And I think Dan gave an excellent talk on the potential utility of RNA-based medicines. And now we also have to show the picture of baby KJ, because, as after TTR, this is the next thing. We're still, very relevant. But how does a lipid nanoparticle work? So before I go to the lung, I just wanted to give a little bit of hint, and, we'll be revising this sort of manuscript, and then I can talk a little more on that. But how do exactly lipid nanoparticles work, especially in vivo, is something that we still are trying to figure out. I think Dan has done some great work on vitronectin. ApoE is clear, but once you take a lipid nanoparticle, it gets internalized inside the cell. It then perhaps breaks apart, and ionizable lipid, which now from neutral becomes positively charged, gets sort of interacts with the negatively charged membrane, and you see an endosomal escape. And currently, the current hypothesis, which was again given 30 years ago or so by Peter Cullis's group, that these lipid nanoparticles, actually the ionizable lipid interacts with the negative charge of the endosomal membrane, and that forms something called a hexagonal phase, which then results in escape from, causes a membrane destabilization, and then escape sort of happens. So currently, this is the theory that lipid nanoparticles, you inject IV, and these are just for the liver. They then shed off PEG, which then interacts with the LDL receptor. And basically, there are other receptors too. We have found certain LNPs that are very ApoE specific, but then they'll still enter the cells without LDL. So there are other mechanisms that we still do not understand. Once these get inside the cell and internalized, they then start breaking apart. And we have done, and others have done, a lot of work understanding the role of ionizable lipids, sterols. And what I had worked in Bob Lange's lab and identified, and Dan Anderson, that this protein called Niemann-Pick type C1 actually recycles some of these particles out. And the escape for siRNA in this case is less than 2%. So they are very effective, but a lot of these particles are not escaping out of the endosomal membrane. I think we have become better. The new SM102, Moderna said it's like around 10% or so. And how do we then assess endosomal escape? The way how we assess endosomal escape, and these were papers that again came in Nature Biotech from Judy Lieberman's lab and Tom Kirkhausen, which suggested that you can take these lipid nanoparticles and then you see a breakage and autophagy becomes highly relevant, and then you see an escape. So these are like labeled siRNA that you see an escape from. And again, large part of these lipids are stuck in endosomal environment. Now we have made the next generation that you see now are biodegradable ones. So hopefully we do not see that much accumulation, and that causes a lot of immune problems. This is a paper that we had published, but these were, again, this was the original paper that suggested galectin-8 is sort of a marker for an endosomal escape. So if you see here, if you take a lipid nanoparticle, these are cells with galectin-8, just looking in the cytosolic environment. With the traditional LNP, you'll see these small punctate structures. That means an endosome is breaking apart. And with cholesterol analogs in vitro, you see a massive sort of vesicular structures formed by galectin now binding to the endosomal membrane. So again, this started to be used as a marker. But the challenge has been for a long time in our field that in vitro stuff doesn't correlate to in vivo. So then how do you predict using all this that this actually works in vivo as well? This is the mechanism. There was another work, again, these are all decade-old work by Marino Zeriel's lab. What he did was he basically with an alnylam took a conjugated gold particle with an siRNA in it, and then put that in a lipid nanoparticle. So these are LNPs with gold in them, injected IV, and then looked for how much cytosolic gold he finds using cryo-EM. So very tough way to do this, but it was found that the escape is around 1 to 2% of siRNA. The challenge with this is no one has done this since again, because this is a very resource-intensive method. So how do we go from understanding from cells to, is there a rapid way, and I've been trying to develop this for 4 or 5 years now, is there a rapid way that we can assess endosomal escape? And more studies will be coming out next, but I just wanted to, and this is the first time I'm talking about it, give you a couple of slides on how we are doing this. So what we started to do was we started to utilize this lysotag mice, which has a LOXP center to it, and it has a marker for lysosomes. So then you generate an animal model by breeding these two animals and getting a constitutively active lysosome. Then what we did was we took a lipid nanoparticle, put a barcode in it, injected it in the animals, and that at different time points took out the whole liver. Now we isolated the lysosomes from it. Now it's a rapid way. So you can start isolating in 30 minutes, 1 hour, 2 hours, 5 hours, to 24 hours. And then what we did next was we basically took the cell lysate, and then once we isolated the lysosomes, and again, this lysosome has a barcode in it, we then basically assessed how much was in the sort of the flow through of without the lysosomes and how much is inside the lysosome of a barcode. And then from that, we could assess a rapid way of assessing sort of the endosomal escape. And we found with some of our LNPs, we see that the escape is around 9, the escape happens very quickly within sort of 30 minutes or so. And then it sort of tapers down. And we've still made like area under the curves around it. And what we did next was we took these lysosomes and did proteomics on it. And from that proteomics, we understood, is there a difference in the lysosomal proteome? And again, we find a huge difference between the lysosomal proteomes of the LNP treated versus the non-treated. Again, this is the liver. Hopefully, we'll go to the lung next. And we have other LNPs that we have tested, and more would come out as we revise this manuscript. And I can talk about this in more detail. But I just wanted to talk a little bit about, can we move from, can we start understanding in a much more deeper way, especially the endosomal processes in vivo? And I think this is one of the way that we are thinking of it. Now moving to the lungs. We have been involved in delivery to the lungs, and I think Dan has done just a phenomenal job. But in 2018, with a collaborator of mine and also the support from the CF Foundation, we basically used CFTR mRNA in the nasal cavity of a CFTR knockout animal, and we found that you can in fact have the CFTR stimulation response that lasts for a few days. And again, we published this, but again, this was in the nasal cavity. So from then on, the idea became like, can we make an inhalable LNPs? And now I know that there are so many clinical trials that are happening. But the challenge we see, again, fundamentally, is that if you take a usual lipid nanoparticle and put that in the sort of vibrating mesh, what you find is that the size increases and there's a change in the LNP structure after nebulization. So, and you also compromise cargo. So one thing you can do is you can change the cargo or the LNP formulation. The other thing that we started to do, and this was with a company called Rare, which I helped start, where using sort of the basic principles of the semiconductor industry, you could utilize a way to use a microfluidic platform to dole out a shear-free LNP. So how this works is that you basically have these microfluidic chips, and this is a digital microfluidic platform. It has different nozzles in it. And, to be fair, these kind of chips are used in the inkjet industry. So for printing, the liquid, the red, green dyes are flown through and they are nebulized and vaporized. So you can use the same phenomena of microfluidics. And using a little bit of heat, what happens is it forms a bubble, and you have different nozzles here. So the ejection time is around 10,000 times per second. And we can control how much, very reasonably, how much the size of the vapor is. And we have published this work in before. And what you can do is also, since this is a cartridge, you can put multiple drugs. You can put an LNP, you can put ivacaftor on the side, and you can control what you give to a patient. And again, this is sort of one example of how the sort of microfluidics uses to aerosolize some of these particles. And what we find is that if with vibrating mesh nebulizers, you see a higher size. But with these new devices, we see that you can control the size. You could further also find that there is no loss of mRNA, but then there is also no fusion. So you are taking two particles together. Sometimes the worry is that they fuse together. But since, again, this is a shear-free kind of environment, a student of mine did FRET analysis to find that there is no fusion happening between multiple particles as we do it. Again, if you use a vibrating mesh, you see these large structures, but with microfluidic platform, even after nebulization, you get these nice pristine particles, which again are good in transfection. You can then take the cartridge, and basically what we did was we took the cartridge, and again, this is digitally controlled, so you can control with the breathing how much dose you give. But the challenge there is then you currently, and that's again a big challenge, you use the whole chamber and the whole mouse is covered with, is wet, is like hiding like from each other. So you lose a lot, but of course we saw a good, expression in the lungs. But that's the beauty of microchips. They can get smaller. And so recently we have actually used a microchip that is much more smaller, same kind of principle. But in this case, now we have put lipid nanoparticles with different barcodes around like 30 or 40, put that on the nasal or mouth of an animal. Again, you see expression, and again, these are less expressive because there are multiple particles here. Then again, using barcoding, we have identified, so these sort of 30 particles are given in like 3 animals or 4 animals, and then we looked for which particle seems to best go inside the lung. From that identification, we moved into validation of it, and we identified certain formulations that actually are hit and are better than the standard SM102 formulations. Then what we did next was we took, again, a Cree-based model, but the difference here is that this already is red, so you see all the cells in red, but once you give recombinase, it becomes green in color. So in the mouse trachea, after using this sort of nebulizer method, these are the sort of transfections that we see. Again, green is red converts to green, and then you can look at like good sort of expression in cells. And this is again just data a couple of months ago. So we are still working on finding what cell types and things like that. We have looked at the airways, and again we see a very nice conversion of the red to green. So it's moving well. We have also done flow cytometry, and again I'll not go more deep in it, but we do find that there is a change in tropism, and we find better epithelial cell transfection with some of our LNPs as well. This sort of device, now we have the capability to actually use it even in humans, but we have moved to non-human primates. We have dosed at least 3 non-human primates by now, putting again barcoding particles as well as individual particles in the NHPs, and we have some preliminary data there as well. This is just one example of that miniature platform now you can use for a mouse in a monkey, and therefore you can use it in a human as well. But you'll have to wait for the NHP data. Thanks to DARPA, we have been pushing this to multiple NHPs at this point. So hopefully by next time I can disclose some of that data to you. We have now, again, thanks to the CF Foundation and NIH, ways to engineer mRNA as well. So with Liang Huang, a computational biologist, we have identified using AI codon optimized mRNA that can even also last longer. So that study is sort of ongoing as well. Finally, I just also want to talk a little bit about another direction that our lab has done. And this is with the work with Marcus Grompe, who's a well-known authority in the pancreas. And Marcus came to me and said, I want to try this intraductal injection to the liver. And I told him that liver is not a problem. But he's like, I can do ducts as well as pancreas. And this sort of method of localized intraductal injection is done in humans as well. So I said, why don't we even try pancreas? And I think this could have implications for CF as well. But I just wanted to share that localized delivery in the lung is one thing, but you can also do it in the pancreas. And so what he does is he sort of blocks, the clamps down, and then passes a needle through and does an intraductal injection, which is again towards the pancreas. Again, there can be some fluid that goes into the liver as well. We have tested this with LNPs, and again the same model. Red indicates no activity, and then green indicates that sort of editing sort of has happened. Most of the expression with the standard LNPs that we are seeing is in the acinar cells here. Around, we have I think 20-30% of the whole pancreas. You don't see much in the islets, but with some modifications, you have started to see something in the islets as well. Again, these are some high-res imaging where I think they got excited because they were seeing something in the islets as well after this, sort of direct ductal injection into the pancreas. So what I'm trying to say is that it's great, and I think there'll be talk by Alex where IV injection can go to broad distributions, but there is so much to be done also on local administration. And yes, there are changes that you can do with the formulation, but there are also changes with the device and techniques that might have clinical relevance and might move very quickly into the clinic as well. We have done this with base editing. We have done intraductal injection in a Loom A mouse. And again, this mouse, if there is base editing, it becomes active for luciferase. And basically what we find is that the luciferase activity can last up till 5 weeks. We're still working on what cell types are we editing using this method. Finally, as much as I love LNPs, this work we have published where you could even try modifications of the lipids with polymers and utilizing this UGI modifications, we were able to make around 150 different polymeric lipid complexes, which again had capability. And again, we have published this work where you can deliver it to the lungs, and you could actually also deliver CFTR mRNA into the lungs. You could do nasal potential difference to see that mRNA works as well. So with that, I think I'll be happy to take any questions. This work was done in collaboration with multiple groups. My grad student Anthony is the one developing the in vivo endosomal escape methods, Namrata for the lungs, and Deepak for the pancreas. I have long-term collaboration with Kelvin McDonald at OHSU for the CF. Thanks again to the CF Foundation. It has been quite a ride working with the foundation and other funding partners. And with that, I think I have enough time for questions.
It's a very nice talk. So I have some comments just about the endosomal escape of ionizable lipids, just for the purpose of a discussion. I know you showed very nicely. I think the shape of the lipids is super critical for the endosomal escape. So you showed that some of those corn-like structures, they're actually good for lamellar to hexagonal transition process, but those are actually a slow process. The key actually, you have to have a large wedge structure like those used for COVID-19. You have a branched and also multi-tailed structure like Dan showed earlier. So this actually incorporate into this lipid bilayer, actually cause the thinning of the lipid bilayer, actually weaks the lipid bilayer. I could push the particles out. So this been actually showed with this COVID-19 vaccine lipids. So we just published a paper to summarize actually detailed the mechanism of how ionizable lipid nanoparticles promote the endosomal escape. I think the key is the structure, the shape of the lipids. So I think that's actually been shown by many publications. So,
yep, yeah. I agree with you. I think, Rong, I've also looked at, I think, one of your papers of H2. I think this was 2008 or so. So I recognize that work as well. You're right. I think the branching is helping. I think more and more people, you can call them different names. Some can call them like rocket shuttle or whatever. But these are all branched lipids. And the question, though, is it what is happening in vivo?
Yeah, that's different story. Yeah.
Everything is in vitro. We are seeing better endosomal escape. God help us. I mean, is it real? Like, what happens if you block? And this paper we have also created another animal model to block late endosomal biogenesis and look at escape. So is this an uptake phenomenon, an escape phenomenon, and where that escape is happening? I completely agree with you that branching is definitely having an effect. But can we do some studies to understand mechanistically in vivo? I think that...
that's different story. Uptaking and endosomal escape are different topics. It has different challenges. So just for the purpose of endosomal escape, I think the shape is critical. And you have this laminar version of lipid nanoparticles like MC3, and a non-laminar version of lipids like COVID-19 vaccines and the multi-tailed vaccines. Those are different. So
I agree with you.
Okay, good. Thank you.
Thanks, over here.
Okay, so my first question is regarding the endosome escape studies. Have you examined different PEG lipids to see if the PEG lipids can impact endosome escape? The reason I'm asking is in our lab, and we have made breakthrough in the PEG lipids. And by optimizing different PEG lipids, we can not only increase the stability for up to 2 years at room temperature, but also more importantly, we found that different PEG lipids can also impact endosome escape?
That's a great point. So we've also worked with like a lot of different PEG variants. We do see differences in the tropism as well as efficacy sort of in RNA delivery, and not just PEGs, but also other variants. So there is definitely something happening there. For endosomal escape, I think the first thing, and again, like this is 3 or 4 years of work going on, we have focused on ionizable lipids and a variety of them. So, I showed you just one example, but we have now 4 or 5 different known lipids, new lipids that we have tested and looked for how does the kinetics of uptake change for all these particles. Because the other thing is what we are also seeing that the pharmacokinetics of these LNPs might also be different. And therefore, how do you relate it to endosomal escape? Again, in vivo is different as well. So again, that story is part of this too. So I've just shown a hint of it. But yes, I think once this goes out, I think hopefully these sort of tools can be used by the community to check for PEG, for cholesterol, for all these other things. But definitely PEG is having some impact in redistribution for sure. So whether it's for endosomal escape, I don't know, but that needs to be tested.
Great. So the second question is regarding your inhalation LNPs.
Right.
And you have AI-guided program to improve stability and potency. So as to stability, have you performed long-term stability studies, for example, at 4-degree and versus room temperature, and how are the results?
Yeah, so that's also actively going on. That's part of our studies, whether, in the cartridge, how long the stability remains, how do we give the cartridge to people. So we have done a lot of studies adding excipients to it, but we have not done I don't think I have the right sort of solution to the stability issue of LNPs at this point. I think they still need to be stored in -80 or something like that with some cryoprotectants or some other buffers. Maybe there are certain companies who have been able to store them at 4 degrees, but I think that's still work in progress.
We can discuss afterwards because sure. Ours can store at room temperature for up to 2 years.
Perfect.
With mRNA.
Great.
So my last question is very.
I'm sorry. I think
we need to move on.
Yeah, we need to move on. But I really appreciate the questions. But please do come back with them for the panel discussion. Thanks. So I'd like now to introduce our next speaker, Alex Piotrowski-Daspit. Thanks.
Great.
So over to you, Alex.
Awesome. Thank you so much. So as I mentioned, I'm a new-ish, I think I can still call myself new, assistant professor at Michigan. I started my lab about 2.5 years ago, and I changed my title slightly to kind of encompass a lot of the work that we're starting to do in my lab. So my new title is Dual Strategies for Cell-Specific Delivery of Genetic Therapies. And I have good news for you. I am not going to talk about LNPs. But yes, so here are my disclosures. So I think, a lot of my intro slides, the other speakers have covered really beautifully, but I, maybe just to briefly summarize. So we've heard at this meeting that there are a lot of really new, exciting genetic therapies that come in a variety of shapes and sizes. But one thing that, in terms of nucleic acid therapeutics seems to be sort of a common feature is that these need some sort of a way to get into cells, right? So these can be degraded if they're not somehow protected, and then also we would ideally have a way to sort of guide them to where they need to go into the body, right? And so my lab solution to this is polymer-based delivery vehicles. So again, we've heard a lot about this, right? So there's the things are really stacked up against us, right? So there's a lot of barriers to delivery in vivo, and I like to think about this in terms of sort of different length scales. And I think, this schematic is really addressing systemic intravenous delivery, but I think a lot of these aspects also apply to local delivery, right? So in any case, whenever you're going in vivo, there are barriers. So if we think about the whole organism level, we think about sort of the immune system, we think about toxicity, we think about accumulation in target versus non-target tissues, right? We think about the ability for the nanoparticles to stick around, to hopefully have enough time to accumulate in target tissues. So all of these factors, right? And then even within an organ or tissue, right, there could be a heterogeneity in terms of local distribution. You can have differences in vascular permeability and extravasation. Again, this is all kind of in the context of nanoparticle delivery, right? And then we heard beautifully from Gaurav about the challenges of endosomal escape, right? So once you actually get to the ideal target cell type, there's additional barriers there, right? So there's all these challenges that we need to overcome. And I just also want to make a plug that sort of the model systems in which we study and develop our nanomedicines really matter, right? So two-dimensional cell cultures are just not predictive of what happens in vivo. I mean, James Dahlman's shown this, others have shown this, we've shown this. And so we really need to be careful about the model systems that we use. I also want to sort of take us a little bit down to the fundamentals. So this session is on targeting, right? And so when you think about targeting, or at least when I think about targeting, I think about sort of the gold standard, sort of, and you decorate a nanoparticle with an antibody on the surface, right? But let's think about what that looks like in vitro or in cell culture versus in vivo, right? And what are the differences here, right? So, you know, in vitro cell culture system is at equilibrium. In the human body, it's a very dynamic and non-equilibrium system. It's also much more highly competitive, and we see much more non-specific binding. And also in cell culture, you know, we can have really well-defined and controllable concentrations, but in the body, once you inject nanoparticles in vivo, you have no idea really. And then, you know, the other important thing too is sort of accessibility of the target, right? And so I'm an engineer, so I like to think about life in terms of differential equations. I'm just kidding, but I think it can really help us to navigate and understand some of the important parameters to play with here, right? So if we think about a nanoparticle, say, trying to interact with its target cell type, so trying to bind to the surface of the target cell type and sort of initiate that internalization and hopefully subsequent endosomal escape. So we can think about describing the rate of nanoparticle binding as a function of concentration of sort of target cells in sort of the vicinity, the concentration of nanoparticles available in that area, and then also, you know, a little bit more sort of specific kinetics on sort of K on and K off of antibody binding. But I think really for my talk, I kind of want to focus on sort of step one here, right? So we want to think about accessing target cell types. So how can we design materials that can when they come into contact with their intended targets, how can we optimize binding of that? But then also, how can we really think about sort of modulating sort of the amount of nanoparticles that are available in circulation, right? So I see this sort of as a twofold problem, right? So hence the dual strategy. So first, we can think about enhancing delivery vehicle affinity for a particular target tissue or cell type, right? So we heard from Dan about really the neat capabilities of endogenous targeting of the sort LNPs, right? So the sort of a basic tissue tropism of these LNPs can direct delivery to different tissues. We can also think about sort of this more targeted version, right? So where, again, you might have sort of targeting moieties on the surface of your nanoparticle that have an affinity for a particular cell type even. But on the other hand, I think it's also really important to think about avoiding delivery to off-target tissues and cells, right? And in the second part of my talk, I really want to talk about avoiding sequestration by the mononuclear phagocytic system, which I think is really, in my view, the first primary barrier to intravenous delivery. And I think actually ties in nicely with maybe some of Dan's results, so we'll see. So I mentioned that my lab works with polymers. I won't belabor the point here too much, but essentially these can be very similar to lipid-based systems. We have a range of chemistries that we can play with, right? We can sort of design materials with different chemical properties. So our particular class of materials is polyamine coesters. So they're a mildly cationic material due to this tertiary amine group in one of our primary monomers. But we can swap in different chemistries to sort of further functionalize or further alter our polymer chemistry. We can also control the polymer chain length, right, molecular weight. And then we also have different formulation strategies by which we can, for example, alter the size of nanoparticles that can be produced. So there's it's a very tunable system, right? Other benefits are these materials are biodegradable. Because we have a mild cationic charge, there's low toxicity. We tend to exhibit pretty high nucleic acid loading. And again, as I just discussed, we can really modify chemical composition and also physical properties. So we can create a library of materials, and we've shown that these can be effective for delivery of a range of nucleic acids, both in cell culture systems and in vivo. But again, back to this fundamental question. So how can we achieve in vivo delivery to the target cells? So the first part of my talk is going to be more on sort of specific cell type targeting. For example, antibody-mediated targeting, right? So we can produce these materials that allow functionalization of antibodies to the surface of our nanoparticles, and we can see really nice targeted uptake, for example, in W1282X HBEs in a 2D cell culture system. But again, the caveat is that this is not necessarily representative of what happens in vivo. So more on that later. So we can also look at different target cell types. So if you remember sort of the fundamentals, right, in order for a targeting therapy to work, the nanoparticle has to be able to come into contact with its intended target. So one way that we can do this in vivo in the bloodstream is by looking at cells that are present there. So for example, we've shown that we're able to target CD4 positive cells in peripheral blood from humans, and we're also able to do this in vivo in a rat model, right? So in this case, our targeted nanoparticles have access to those antigens that they're able to bind to, right? And so next, I want to highlight the work of one of my fabulous first PhD students in my lab, Owen, who actually spoke in a previous session. So I won't, belabor this, but I just want to give you some highlights. So we're working on an alternative targeting moiety to antibodies. So antibodies also have a lot of other factors that are maybe less desirable, right? So the FC region of antibodies can be really immunogenic. And we also want to think about targeting moieties that maybe don't obscure or totally obliterate the inherent properties of our polymer material. So can we sort of take advantage of natural tissue tropism based on polymer chemistry, but then also maybe give a little boost of targeting? So we're looking at these engineered short peptides that bind to EpCAM. And so what Owen has found is that if we compare these to sort of the gold standard antibody nanoparticles, antibodies win all the time in cell culture, which is not surprising. So antibodies also tend to have a higher binding affinity. I think it's twofold higher. But again, this is in 2D cell culture, so it's not necessarily what we can expect in vivo. And in fact, it's not what we see. So we've really tried to push towards evaluating our nanoparticles in more physiologically relevant model systems. So for example, we have been using human precision cut lung slices, where we've been fortunate to receive donor lung samples from the Gift of Life Michigan Foundation that are not suitable for transplant. And we're able to slice these with a vibrotome and culture them, and then assess our nanoparticles here. And so we've confirmed that these PCLS tissues express EpCAM robustly, which is great because this is the proof of concept target that we're going after. And here's where it gets really interesting. So it turns out that in this ex vivo culture model system, antibodies don't really do very well. They're actually not much better than the isotype control antibodies. It turns out our peptide nanoparticles are much better in this more physiologically relevant system. It's still not perfect, right? It's still not a whole human lung, but we're kind of excited about this. And I just wanted to make a quick point. I think Finn Hawkins mentioned at an earlier session that we also, in these sort of complex tissues, we really need to look at sort of, in-depth confocal microscopy and staining of different cell types, and so that flow cytometry maybe doesn't necessarily capture all of the data, right? Because it can be really hard to sort of fully digest all these tissues and retain all of the cell types. And so our approach has been really to quantify our targeting ability via imaging and then quantitative image analysis. So we're excited about this, and we're hoping to apply this to multiple applications, including, of course, CF. All right, so let's see. So the next slide, right? So there are ways that we can enhance delivery to a particular target cell type, but then how do we avoid delivery to off-target tissues and cells, and how do we avoid sequestration by the MPS? So the mononuclear phagocytic system is a part of the immune system. It's a collection of sort of different phagocytic cells, primarily monocytes and macrophages. And these are really primarily responsible for the sort of first pass foreign body particulate response in the body, right? So here on the right, I'm showing just some of our polymer nanoparticles in red, and then I'm showing Kupffer cells or macrophages in the liver. So sorry, this is the liver section in green, and you see almost complete co-localization of our polymer nanoparticles in the Kupffer cell. So that's well known. It happens to almost all non-viral nanoparticles, right? So what can we do about this? Just more evidence, right? So we've seen in a variety of different polymer nanoparticle systems that liver accumulation seems to be primarily responsible for accumulation. And if we look specifically via flow cytometry in bulk liver cells, so these are, we studied in vivo delivery of a subset of our PACE nanoparticle chemistries. And so, and then we really wanted to understand what are the cell types in the liver that are responsible, and no surprise, that's the 480 positive macrophages. So what can we do about this? So last year we published a paper in Nature Communications. So this was sort of the last paper for my postdoc in Mark Saltzman's lab, and really sort of driving some of the new directions in my own lab. So we thought, okay, well, what if we can temporarily occupy these macrophages, or sort of blockade them, such that we can subsequently deliver something else that has a better chance of getting to other tissues besides the liver. And so we tried a variety of different strategies, and lo and behold, we actually found that we can boost biodistribution. And no surprise, if we completely eliminate or deplete macrophages using claudinate liposomes, which are well known in the literature, we see the most widespread distribution. But if we use some of these more mild therapies, such as intralipid, which is actually a lipid emulsion that is given to CF patients, interestingly, or cargo-free nanoparticles composed of another polymer, polylactic coglycolic acid, we can also boost biodistribution. And so this really it inspired the project of my other graduate student, Peyton, where she's really trying to engineer these cargo-free nanoparticles to intentionally go to the liver and occupy these phagocytic cells. And we're looking at a lot of factors like nanoparticle size and sort of the surfactant that we use to formulate these nanoparticles, and how that affects this sort of phagocytosis. And so the idea is to pre-treat with this sort of, we like to call them decoy formulations, such that you can get more widespread delivery after. So I'll tell you a little bit about her work. So she started in cell culture. So we looked at therapeutic nanoparticle uptake in primary bone marrow-derived macrophages from mouse models. And in this case, what you're looking at is, so we pre-delivered empty nanoparticles, so the decoy nanoparticles on the bottom here. And then after that, we delivered a diluted, what we're calling a therapeutic surrogate formulation. So what we're looking at is uptake by these macrophages, right? So the higher number is technically worse, right? So if you look at DID nanoparticles only, you see a high level of uptake, right? But then once you start to pre-treat, the amount of therapeutic nanoparticle that gets phagocytosed is going down, right? And we looked at a number of different parameters, like the size of the decoy, concentration, and then PVA and PIMA are two different surfactants that we can use. So this is starting to be exciting, right? And we're like, okay, so can we translate some of this to in vivo systems? And so this is some of the work that Peyton has done, and it turns out we actually can. So if you pre-treat with these decoys, you can actually pretty significantly shift biodistribution of the same nanoparticle type. So this is really exciting, and I think we can also apply this to not only our polymer-based nanoparticles, but hopefully to LNPs or any other non-viral strategies. So here again, you're looking at therapeutic nanoparticle only, again, primarily accumulation in the liver, but once we do these pre-treatments, we start to see a shift. And if we look at flow cytometry quantification, I think the effects become a little bit more clear. So with our different decoy formulations, we're actually reducing the amount of nanoparticles present in the liver. And then this one particular formulation, decoy formulation, results in a really significant boost of delivery to the lung. And we're now exploring sort of what cell types we're able to get into. But from some of my prior work, we've seen that we're able to get into endothelial cells and epithelial cells. So we're really excited about this. And I think maybe this, I don't know, the Martin Mentz's question to Dan about sort of timing, delivery, I don't know, it seems like there might be something synergistic here, right? So if you pre-deliver something, you might boost delivery to the lung, at least in our context. So we're really excited about this. So to conclude, I hope I've convinced you that, I think all of us have convinced you that delivery to target tissues in vivo is a key barrier for clinical translation of therapeutics, at least of non-viral carriers, that we can surface functionalize with targeting moieties, but that sort of we have to be careful with this, and we have to make sure that we're screening in relevant model systems to identify our lead candidates, and then also that biodistribution and phagocytic clearance can be modulated to increase accumulation in key tissues and cell types. And with that, I'd like to thank our lab, and especially Owen and Peyton, who did the work in the first projects from my lab, as well as our funding sources. So Emily's Entourage, Pharma Foundation, and Cystic Fibrosis Foundation in particular. And I know this is the last session of the conference, but if you're interested, these are the other presentations from my lab. So I'll be happy to take questions.
Okay, thanks, John. Go ahead.
Yeah, that was great, Alex. Two things. So why is it that the antibodies didn't work, but the peptides did? Is it based on size, you think, or is it something different than that?
Yeah, so I gave a very small snippet of the story, so Owen gave the full story earlier. But we think what's happening is that, so the particles that we use in this case are pegylated, and we think that the smaller size of targeting moiety and also not complete surface functionalization retains some of the stealth properties of the PEG, and so we're able to get deeper tissue penetration. So yeah, we've been looking sort of, at various depths of the PCLS cultures, and we see more peptide nanoparticle signal deeper in, which is interesting. But yeah, we're still evaluating.
And then your decoy thing is fascinating. It's really cool. And so, but you keep on saying it's for non-viral. Do you think that you could use a decoy method
Possibly,
for
yeah.
AAV?
Yeah.
Yeah.
That would be cool.
Yeah.
Yeah. All right.
I had a question about the EpCAM targeting. So what was your starting point for the rationale for targeting? This is the epithelial cell adhesion molecule.
Yeah.
What's the so this protein? What is its distribution on the cell? Is it involved in uptake into epithelial cells?
Yeah, that's a great question. To be honest, we used it as a proof of concept. So in our staining of 16-HBE cells and also these human precision cut lens slices, we see that EpCAM expression is pretty ubiquitous across the surface. So we thought it would be a good proof of concept. So we know that sort of the abundance of what we're trying to target is there, right? So we're not playing with sort of like hard to get targets. So just as proof of concept for this technology, that was our rationale. But I think, as we get more and more refined in sort of our therapeutic strategies, and as we learn more about which cells are important, for example, in the context of CF, in terms of CF expression, I think our hope is to then refine these strategies. And yeah, did I answer your question? Hopefully.
Yeah, I think so. Yeah. Marie?
Hey, sorry. Great talk. When you think about your decoy strategy, do you have any idea of what the shortest time frame is between decoy and delivery of your true cargo?
Yeah, that's a great question. So we've just started to play around with that. I think, we do see some effect when we co-administer, actually. But it seems, it depends on the decoy a little bit, but we seem to see the most benefit between 24 and 48 hours prior. Yeah.
Hey, really wonderful work. I'm always excited to see your lab's research.
Oh, thank you.
So I have a very simple question about the decoy nanoparticles. Is there preferential uptake for, what's the sweet spot? Is it like 400 nanometers? Because it almost seemed in one of your plots that you showed, it drops off at 500 nanometers, but under that it's...
Yeah, and that's an excellent question. So I think we're just scratching the surface. And the other thing we think is that I think the combination of choice of decoy and choice of therapeutic is going to be really important, right? So we're also going to look at, okay, so we really, varied sort of the decoy properties, but then what happens if you vary sort of the therapeutic properties? And I think, unfortunately, I don't know that there's going to be sort of one solution fits all, but it might really depend on that combination pair. So we don't know exactly, but good question.
Okay, thank you.
Okay. All right, so our last speaker is my co-chair, Stephen Hart.
Thanks, Alex. Oh, brilliant. Okay, thanks for the introduction. I have no declarations. So I'm going to talk about another kind of delivery system. I think it tells you something. This is a very vibrant space, the non-viral delivery system. Everybody has their own formulation and type of nanoparticle. And we're no different. We have our own as well. So I'm going to tell you a bit about the nanoparticles that we work with, which are formulations of lipids and peptides. And I'm going to talk about some of our work delivering CFTR messenger RNA and the opportunities we see for improving expression. So we've recently heard from Arcturus some very promising results from their clinical trial on CFTR messenger RNA delivered with lipid nanoparticles. For those of you not in the field, CFTR messenger RNA has, we think, improved potential for a kind of a gene therapy in that it's a more potent molecule than previously attempted plasmid DNA approaches to gene therapy. But it's a very fragile molecule, so it has to be packaged with a carrier to get into the cells. And it's transient. The messenger RNA turns over quite rapidly, so it's going to have to be repeatedly delivered. So there are various opportunities in that pathway to optimize transfection from the LNP itself to the messenger RNA and to, as I'm going to talk about, stabilization of the protein. So this is the formulation that we work with and have done for many years now. So it comprises lipids. So this was one of our starting lipids. So I'm just going to show this one for now. Similar to DOTAB that we've already heard about. This is DOTMA. So it has an 18 carbon tail and a diether linkage and a amino charged head group. And it's formulated with DOPE. Which is a very potent lipid for endosomal escape, which is, of course, one of the main barriers to delivery. This lipid has been around for many years and is still used in the lab as lipofectin for transfection experiments. But we formulate this with another important component, which is a peptide. And what we're looking for with the peptide is to provide additional packaging capacity, which is from our 16 lysine domain, and importantly, a targeting motif at the other end. And this is typically a short sequence of it can be just 7 amino acids. We've even done as small as 3 amino acids. So peptides are fantastic, very diverse molecules can encompass a range of functionalities, especially in targeting. So if you formulate all this together with a nucleic acid at the right ratios and in the right order, you get these kind of nanoparticles. At least this is how we envisage them, with the peptide displaying its targeting motif on the surface, the lipids sort of jumbled up in between, and the mRNA or other nucleic acids packaged. It's difficult to be very precise about how this happens, but we have some insights into the structure of the nanoparticles. So if you start with a liposome, this is by cryo-EM, you see classic lipid bilayer structure. This is a liposome with nothing in it, like DOTMA dope. If you package messenger RNA with a liposome, you now see these, you can see a smaller particle, but also you can see like bilayer structures where the mRNA is packaged in between layers of lipid. It's kind of a fingerprint structure. Then we add our peptide component, and it's all change again. We completely lose the bilayer structure, detectable by this method anyway, and it now seems to be a rather featureless, amorphous mass in the middle. And we've shown by functional testing that there are targeting motifs on the cell surface. So this is the particle that we work with. So just briefly, we've heard a lot about endocytosis and the challenges of delivery. But the reason why plasmid DNA mRNA is much better than plasmid DNA is that we miss out one of the key barriers to transfection, which is uptake into the nucleus. So of course, messenger RNA is released into the cytoplasm where it can engage with the translational machinery in that location without having to get into the nucleus. So messenger RNA is good. Do our nanoparticles work in vitro, in vivo? So we've done in the past oropharyngeal installation experiments delivering luciferase. This is immunostaining for luciferase expression, and this is even with a plasmid DNA. We can get very good transfection in the mouse lung. These are mice that were nebulized. I think our nanoparticles are quite stable to nebulization. This was even done with a jet nebulizer because of the additional peptide component. We've also delivered to large animal models, including pigs, by nebulization. So it has promise for in vivo uses, but the mice do not represent the challenges that we face in the human lung. So we still feel there's obviously a great need for improvement of the various steps in the transfection pathway. So here, this is in vivo comparison of plasmid DNA, PCI loop. This is a CMV promoter, very powerful promoter driving firefly luciferase. We deliver the same amount of messenger RNA, and there's a more than 2 log-fold difference in luciferase expression. So that just illustrates the power of messenger RNA. There's a very strong linear dose response as well. So when we do immunostaining, I don't know if you can see very clearly, but there's abundant staining in the airways. And very importantly, very little evidence of inflammation. So this was a GFP messenger RNA treated mouse. This was a luciferase mRNA treated mouse. And this was the plasmid DNA treated mouse. And you can see these signs of infiltration. But the examiner reported that he couldn't tell the difference between the mRNA treated and the untransfected samples. So it looks very promising from a toxicity point of view. Looking at luciferase expression, which is a very short-lived enzyme, there's a half-life, I think, of about 2 hours. It decays very rapidly. So peak expression is at 24 hours, but by 72 hours, it's more or less getting down to background levels. And there's a half-life. We can estimate the messenger RNA half-life is about 7 hours. So there's people who've done a lot of work, as have we, on stabilizing and improving expression from messenger RNA. But where do we go next? So we thought now to look at the protein itself, CFTR protein. So an interesting feature of translation of endogenous CFTR is that it's a very inefficient process. So it's reported, several papers, only 20 to 30% or so of translated endogenous CFTR makes it to the apical membrane. The rest is degraded due to folding errors. And we're assuming that's going to be the same problem with in vitro transcribed CFTR messenger RNA. So it looks like there's quite a big opportunity here to improve expression by stabilizing protein expressed from the mRNA. the approach that we took, of course, was to look at the correctors to see what if we could affect the stability of CFTR mRNA that way. So we started off, we did the in vitro transcription. This is a wild type CFTR. It's non-codon optimized, and it was also non-chemically modified. So we made this in the laboratory, formulated it into our nanoparticles, and we did various transfections, transfecting basal epithelial cells. These are CF cells, and also air-liquid interface cultures for functional testing. So the nice thing about nanoparticles like this that contain lipids is that there are various compartments for packaging drugs and cofactors. So you have the lipid layer, which is obviously hydrophobic. There are aqueous compartments within, and electrostatic packaging because of the highly charged nature of the nanoparticle. So we prepared our nanoparticles, but during the mixing, we just added VX809 in with the lipids. This is in a DMSO mix. So the VX809 will be incorporated into the nanoparticle and packaged into the hydrophobic compartment within the nanoparticle. And we did our transfection in our CFBE basal cells. And we found, we tried two different formulations. We had two different lipids, but the same peptide, and two different doses of the mRNA. And you can see from this Western blot formed at 24 hours after transfection that our native or in vitro transcribed wild-type CFTR gave expression with band C, band B, but is much stronger with the VX809 treatment, and that was consistent across two doses and in two different formulations. And when we looked at the ratio of band C to band B, in most cases, the proportion of band C was increased in each case. So this looked promising. We then did looked more longitudinal study, just going out to 48 hours and then 7 days to see if this would have effect on the persistence of the protein. So we see expression very early on of CFTR, and you can see clearly that most of it is band B. So this is freshly synthesized CFTR protein. By 8 hours, it's now becoming equally distributed, I would say, between band C and band B. By 24 hours, it's nearly all band C. So what we're interpreting this is that not all of the protein is being converted to band C, but synthesis has stopped because of the decay of the messenger RNA, and the protein that's been successfully made has now been converted into and trafficked to the membrane and fully glycosylated. So this was just VX809. So we tried a few other correctors and one potentiator. So VX661 is an interesting corrector in that it binds to the same pocket in CFTR as VX809 and is now preferred clinically because it has a better toxicity profile. But VX809 is known to have higher affinity for wild-type CFTR. So when we did our Western analysis again, we indeed saw that. So here we have transfection with just a DMSO control. Here we have VX809. So again, we're seeing the strong enhancement by the corrector. 661 is working, but not as strongly. It's almost half of the enhancement that we see with VX809. So that's kind of consistent with what's known about the properties of 661. VX445 was not as good at all. That binds in a completely different part of the protein. But what surprised us enormously was the potentiator VX770 worked almost as well in stabilizing the protein as VX809. And I've been talking to people and reading literature, and I haven't found an explanation yet, but I see Bill's here, so maybe he can help me afterwards. So what's going on with these correctors and the potentiator? So in the same way that obviously they were developed for stabilization of F508 and other folding deficient CFTR molecules, but they're very good also at binding wild-type CFTR. So we're speculating that as the protein, the nascent protein is formed, the TMD1 is one of the first, is quite early in the translational process. It's one of the first domains made. So there's an early opportunity to VX809 to bind to the protein as it's being formed. And that is perhaps stabilizing compared to samples without VX809 and ensuring correct folding. So we don't get, we haven't done the measurements, but instead of only 20 to 30% surviving the processing within the endoplasmic reticulum, now maybe we've got a higher proportion is surviving. So, it would seem we've increased transfection efficiency fourfold by focusing on the protein. As I say, what 770 is doing, I'm not clear. So we've then done functional testing to see if we treat these are CFTR F508 homozygous cells, and we treated them on aliculture. We did in Ussing chamber. So we did the inhibitor and activator study, the classical amiloride forskolin inhibitor. So we see, so looking at the forskolin response, a small response with the combination of 809 and 770. I haven't got the mRNA alone on this graph, unfortunately. There weren't enough wells in the plate for that, but it's very low with the mRNA alone. But when we combine 770, 809 with the CFTR messenger RNA, we now get a very strong forskolin response, at least as good as the wild-type cells, so into normal range. And the response to the inhibitor as well is similarly in the normal range compared to our NHBE cells, and much higher, significantly higher anyway, than the response to the drugs alone. So, in the interest of time, I'll move on to the summary. So what I have shown, I think, or happy to discuss, is that mRNA is a more potent nucleic acid than plasmid DNA for therapy of CF, but challenges of delivery and persistence remain. We don't want to have to be repeating this weekly. So we have a nanoparticle that's shown very good expression in normal mice, but the challenge is to translate this to humans, and we're focusing on the physiological barriers, including mucus penetration, targeting cell types. But here we've focused particularly on stabilization of CFTR by modulators. And we're proposing, I think it's probably something that's going to happen anyway, but I think it's interesting to look at what is the effect of the combination of CFTR messenger RNA with different modulators. Are the modulators that we use for therapy on their own, the same ones that are going to enhance wild-type CFTR in vitro transcribed mRNA? So yeah, there's lots of questions that we have from this study. So what we're planning to do now, so we did these initial studies in F508, but now of course we want to look at cells with nonsense mutations, as these are probably the primary beneficiaries of CFTR messenger RNA. What about using now codon-optimized CFTRs? So there was a great presentation earlier in the conference about codon-optimized CFTRs. So that's going to be an interesting question. Do the modulators interact the same way with codon-optimized CFTR? What is the persistence of correction in the alley with these drugs? Can we get an insight into how often we're going to have to repeat dosing? And how exactly are they working in this context? So we're also, in parallel with this, developing our nanoparticles. We have selection of new targeting peptides we've identified by phage display with cell type specificity. So it would be interesting to look at specific cell types, what the responses are as well. So at that point, I will stop and thank all of my collaborators. Especially I'd like to thank Rahina, who's a postdoc in my group, who did most of the work I've shown you here, and Debbie Baines at St. George's, who does the electrophysiology. So with that, I'll stop and happy to take any questions.
Questions for Steve?
Great. So the idea of being in the specific cells is really exciting. Targeting to the cells that have CFTR expression. Do you think that the match when you get it to different cell types is going to be different? Do you think that some of the issues you're having is that you're putting into a cell type that doesn't express CFTR?
Yeah. So we're, for the Westerns, these are all basal cells.
Yeah.
They're not differentiated cells. So yeah, that's kind of the next step to. I mean, as we've heard, alley cultures are very difficult to transfect. So we're doing the initial donkey work in these easier to transfect cells. But yeah, it's going to be important, I think, to look at responses in different cells that perhaps have higher amounts of, like, the ionocytes, the secretory cells. What's
I mean, it may be better.
Yeah.
Right. It's
Yeah.
not a bad thing. It's going to be better. Yeah.
Yeah.
Hi. So I just wonder, you talk about the potentiator and the correct co-transfector with this mRNA CFTRs. I just wonder, so potentiate effect if you acute addition, I mean, so for example, mix with force clean or first force clean it and then add this potentiate acute the potentiate effect on the CFTR function.
Yeah, well,
test it?
Yeah, so the potentiator, its known property is keeping the gate open longer, isn't it? So, but this is kind of suggesting it's stabilizing newly synthesized protein as well.
Yeah, I just curious because we do find in some cases, so I mean, the potentiate, for example, is a safety MRI after deliver, or some DNA the delivers do find some potentiate, some have tiny bit inhibit the force clean response.
Oh yeah.
So this is why for you this case,
Yes.
I just curious the acute the potentiate, what's going on in you.
So I think yes, I've read that paper, and I think the response that you're talking about was in F508 CFTR, specifically not wild type CFTR. But there are people here who know better than I do about this.
Yeah, because I believe you mentioned it because safety mRNA after the translation, maybe this CFTR is have tiny bit different. Native CFTR folding have some changes because you this CFTR mRNA already do some modified, right? So for these reasons, it will be very interesting for me. Yeah.
Thank you.
Thanks.
Great talk. Thank you so much. I'm Anuja Pandey from CFFT Labs. I know you mentioned about some work with nanoparticles and using phage display for identifying peptides. I don't know if I missed it for this particular design, experimental design that you showed, the heptapeptides, how were they screened for? Was it a particular different method, or did you still use phage display for it?
Yeah. So sorry, I didn't actually show that. I should have shown the sequence. It's not a secret. So that peptide, which is SERS-MNF, if anybody wants to know, it was selected by phage display and has homology to known binders to ECAM, not EPCAM. I don't think they're the same. So that peptide has affinity for airway epithelial cells. Mm-hmm. But that was only panned on a cell line, 16-HBE. So we've been doing further biopanning on differentiated cells. So I think we've got some more selective peptides.
And just to add to that, was it the panning library was something that you all synthesized in-house, or was it a commercial one?
It's the NEB PhD
C7C. Okay, got it. Thank you so much.
Thank you.
Maybe one question.
Okay, thank you. One. So you mentioned that CFTR mRNA therapy, is mRNA expression persistence is very critical for such a therapy.
Yeah.
So have you tested or thought about to screen for different nanoparticles with the slower endosome escape, so that the cells can express lower level of expression, but with persistent expression?
Yeah, not that specifically, but slowing down the whole process, giving the protein more opportunity to fold correctly is something we're thinking about. But slowing down endosomal escape could have a downside as well, in that you might get more degradation in the endosome. So you might get less endosomal escape, as well as slower.
Yeah, the reason I'm asking is not to specifically modify the specific LNP to slow down, just to screen for different LNPs with different properties.
Yes.
The reason I was asking is because we realized that even for mRNA therapies and for different indications, you may need to have different expression levels. For vaccines, you need rapid release and high levels. But for long-term therapeutic, for chronic diseases, you really need a persistent expression.
Yeah.
So by just trying different nanoparticle formulations, we can see a significant difference in the mRNA persistence.
Yeah.
Thank you.
Thank you. Thanks for that.
Awesome. All right. Thank you, Steve.